The findings could have implications for understanding how our eyes help regulate mood, digestion, sleep, and metabolism. NEI and NIMH are part of the National Institutes of Health. Vision begins when light travels into the eye and hits the retina's light-sensing photoreceptors. The photoreceptors transfer signals through several layers of retinal neuron before those signals are sent to the brain. Light also triggers certain non-vision functions, such as controlling how much light enters the eye through the pupil, pupillary light reflex, and regulating the wake-sleep cycle, circadian rhythm. The group investigated the roles of rod photoreceptors, which are sensitive to low light levels, cone photoreceptors, which see color, as well as three types of bipolar cells: rod bipolar cells, on-cone bipolar cells, and off-cone bipolar cells. Bipolar cells then communicate with other neurons in the retina, passing information to the optic nerve and onto the brain. Some mice in the study had no functional connections between rods and bipolar cells, for example, or connections between cones and any bipolar cells, or lacked connections between rod and cone photoreceptors. The researchers compared the mice's responses to visual stimuli while assessing pupillary light responses and monitoring their nocturnal wake-sleep cycle. They determined that while image-forming vision can use rod and cone photoreceptors, as well as all the types of bipolar cells, the same was not true for non-image-forming functions. We were really surprised to find that animals with only bipolar cells can't adjust to changes in the day-night cycle, but can still see and respond to visual events, meaning they have functional image-forming vision. It was really interesting to us that the non-imaging forming functions completely ignore information from the off pathway, said Paulberg. We were equally surprised that rod photoreceptors, which are optimized for low light conditions, were still being used for the pupillary response even when light levels were high. We really thought the rods would be maxed out at that point. Paulberg expects many of these findings in mice will hold true for humans, since the retinal circuitry is similar across mammals. Moving forward, he intends to explore other non-image forming functions of the retina, like mood regulation, and see how else these different retinal circuits are being used. The research was funded by the intramural programs of NEI, NIMH, the National Institute of Dental and Craniofacial Research, and the National Institute of Neurological Diseases and Stroke. Make a difference. Sponsored opportunity StorySource, materials provided by NIH National Eye Institute. Note: content may be edited for style and length. https://ift. TT, ADP0Co7, accessed July 6, 2022. Thanks for watching. Please subscribe my channel for more videos.